give us a talk today.
Thank you. That was incredibly kind introduction. Folks in the back, can you hear me okay? Cool. Yes, thank you guys so much for having me. I'm from all the way down to T-Stops at Kendall, over at the Broad. We're right next to the Whitehead, if you know where that is. There's QR code and short link for the slides. I tend to talk too fast, and there's a lot of resources I want to make sure that you guys are at least aware of. It'll be throughout the talk. You don't have to do it right this second. But just so you know, if there are things like you can try to focus on listening, you can grab the slides later. If you're just here for the pizza, that's also fine. So just question, who has taken an image on a microscope before? Most of the folks. Who has done some sort of image analysis on that image before? Just about everybody. Put your hand down if it's not Fiji. Okay, yeah. So most folks, again, all of us start with Fiji, including myself, and Fiji is great. It's still the absolute first thing that I use when we get new images in the lab. But I'm going to tell you about some of the tools that our group writes, which once you've then sort of done your initial image exploration, hopefully will make your life a little bit easier in terms of sort of creating quantitative analysis, especially high throughput quantitative analysis. So there's going to be sort of three little 10-ish minute subparts to this. How can you analyze lots of images without learning to code? How can you do that with deep learning and also not learn to code? And then how can you do more to take, especially if you're doing things where you have things like multiplex stains, how can you use every single one of those pixels to get really high dimensional, really powerful analyses? For that, you probably have to code a little bit. And again, nothing wrong with learning to code. If it's something that you're interested in doing, totally recommend it. But sometimes you also have other things you're trying to learn or other things you're trying to do. And we're trying to make it so that learning to code is not a minimum requirement for doing great microscopy and therefore great image analysis. Hopefully most people in the room are on board with this idea that in addition to just being beautiful, which they are, when you take an image on a light microscope and a biological image, it can also be pretty informative. And there's things that even as we get, this omics and that omics and metabolomics and proteomics and lots of omics, the ability to do things in live cells over time with multiplexed stains and see how the behavior of molecule changes, not just their expression level, is still something that most of the time you really need a microscope for, especially if you want to do this a few times or you want to do it affordably. So there's still a lot of things that you really need a microscope to do. But the problem that we face in microscopy and in bioimage analysis is historically, bioimage analysis is an add-on. Most of microscopy's history, I've been doing microscopy for 400-ish years, was about this. It was about drawing what you saw and sort of evaluating first what you thought you saw and then writing it down or drawing it. So the historical bias for bioimage analysis is we don't need that. We spent 400 years not doing it. So why on earth would we bother? And hopefully you will not think that by the end of this talk if you do think it now. If you do think it now, it's okay. There's no judgment. But hopefully you will think that less by the end of the talk. Because when you have a digital camera or anything, anything that makes a digital image, you have just an array of numbers. And that's true of any picture you take. That's true of the pictures you take on your smartphone. That's true of this picture of a man taking a picture. These are just arrays of numbers. And so a standard microscope camera these days is 4-ish megapixels, which is 4 million data points. You have 4 million data points every single time you snap that camera. And that's true for multiple channels and multiple z-planes and multiple time points. So you end up with sometimes billions or even trillions of points of information that for the most part don't really get used quantitatively. And this is going to mostly set up the sort of third part of my talk, but I want to introduce this idea now that what we're looking at are not actually pictures, they're arrays. But again, that's not how, as biologists, and again, I'm trained as a biologist, I have no formal coding experience. We're not trained to think of images that way. We're going to go and we're going to optimize our sample prep, and then we're going to go on the microscope, and then maybe we didn't pick the right microscope and the right sample combination, so we're then going to go back to the sample prep, and we're going to just sort of ping back and forth between sample prep and image acquisition and not go further until we're done. And unfortunately, that's a shame, because when you actually then get into what do you want to learn from your information and or from your images and how do you report it, what you might find is there were things that you could have done at the sample preparation and or the imaging stage that would have made your life a lot easier. This whole thing isn't supposed to be a line, it's supposed to be a circle. So we recommend that you think about your image analysis as soon as you start thinking about doing a microscopy experiment. And as soon as you have pilot images with controls, because you took those, right? You did some controls and took some pilots. I know you guys did that. You use that to do a preliminary analysis and see if there are actually things that you need to change at the bench so that your life at the laptop gets a lot easier. So I work at the imaging platform, was started by Ann Carpenter. She is the co-original creator of CellProfiler. She and Shantanu Singh run the Carpenter Singh Lab, and they focus mostly on image bioinformatics. And my team focuses mostly on that. How do you turn an image into numbers in the first place? How do you actually find the biology in an image and turn it into a measurement? And they mostly focus on, once you have a measurement, how do you use that to answer a question? But we all work very closely together. And our whole goal as a team is to turn images into answers. Okay, at least one person in the room, I guarantee, is thinking this. You can see your phenotype just fine. All right, fine, I'm here for the pizza. Here's the problem. Human brains are very bad quantitative engines. Those of you who are in the back might still see this as a color picture. Those of you up front certainly almost certainly saw it that way at first. This is not a color picture. This is a grayscale picture with some lines drawn on top of it. And your brain has very helpfully filled in the missing information to try to help you get a sense of what's going on faster. Great for, surviving this long, but not actually helpful for quantification. So even if you are doing your absolute best to treat your images rigorously and blind the image names before you assess your phenotypes and everything like that, your brain is still lying to you in ways that you don't necessarily always pick up on, in a way that a computer will not be tricked the same way. So yeah, the first 2/3 of the talk, I'm going to talk about what most of you probably do, which is you go into an experiment sort of knowing what is the thing you want to measure. This is the way most biology is done. You might have fused a fluorescent protein onto a thing that you care about, or you might have found a really good antibody for it. I have done this a lot. And at the end, we're going to talk about some other ways to do it, but this is probably what most of you are doing. And I'm going to, again, sort of link you here to a blog post that has sort of the really hardest part about image analysis is knowing when it's good enough and when you're done. And more than any individual tool, understanding the principles of how you do this is going to be way more valuable, because the tools are going to change constantly. You're going to be using different tools 5 years from now than you are today. But if you think about it, when you're doing your microscopy and your image analysis, saying, do you generally agree that the image analysis is finding most of the objects that you detected on the microscope? When it's wrong, are the kinds of errors, a little bit too much stuff detected or a little bit too few stuff detected, are those balanced? Is it about the same? If it's too high in one direction or the other, you can probably fix that. But if you have about the same number of errors of like a little bit too much stuff, a little bit not enough stuff, there's probably reached about the limit of what you can do. Same thing for objects being split in half versus being shoved together. And very importantly, almost always in biology, you're comparing between, a control and a treated, or a patient and a control, things like that. Is the algorithm treating fairly both of those sets of conditions? I can't tell you how heartbreaking it is to spend months trying to chase down what turned out to be a segmentation error in one of your conditions. Actually, I can tell you because I did it in grad school. It's not fun when you then have to go to your boss and be like, actually, that biology wasn't real. My analysis was just bad. It's not a fun day. Avoid that day. Check this first. So how do we actually do this? So this is not a CellProfiler workshop. It's not a CellProfiler pitch. We do run CellProfiler workshops if you ever want those. But mostly I want to show the slide because I want to highlight the really fantastic people over the years. CellProfiler turned 20 last year, so there have been a ton of people involved in it. This is just sort of the people who've been involved in it a lot. But I think part of the reason it stood the test of time, other than ImageJ, it's probably the sort of most stably used software in bioimage analysis, is because for the whole of its history, going back to Anne and Ray, who made the first version, there has been at least one computer scientist, the folks in blue, and at least one biologist, the folks in green, who have worked together to work on the software. And so we get good computer science, but we also get all of the documentation being written by biologists and in language biologists understand, and trying to pivot the tools in the code towards the kinds of experiments biologists are likely to want to do. So we really do try and design it for you guys. Again, I'm going to just give you a quick overview because I think this is a tool that for some of you is the right tool to do your image analysis, but we're not going to belabor this. CellProfiler, why would you use this instead of ImageJ? Well, CellProfiler is at its heart an image analysis workflow tool. For those of you who are comfortable coding and writing ImageJ macros and things like that, great. You're probably good staying with ImageJ unless you have tons and tons and tons of images, because ImageJ has a ton of plugins. Fiji is great. But if you're not super comfortable writing macros and coding, what happens when you figure out a beautiful workflow to find the objects you care about and measure them, and then you want to do it for a second image, for a third image, and for a 300th image or a 3000th? What CellProfiler does is it lets you create an image analysis workflow in what we call a pipeline, where it says, here are individual steps. You should do them for all of my image sets, whether that's 3 or whether that's 3 million, and do them reproducibly over and over again the same way without having to know how to code. In fact, it will even, unless you turn it off, write a copy of the pipeline into your quantitative measurements so that you know right next to the measurements exactly how these measurements were created. Please don't turn that off. People do, but please don't. And so what this allows you to do is you have about 100 or so different modules that you can put in a pipeline in any number that you want and any order that you want. Each module you can then configure. Some of them have very sort of limited numbers of settings. This is the images module. You pretty much just put in images. Not a whole lot of settings here other than being able to filter some out. But if you are confused about what's going on in a module, this is the most important button in CellProfiler. This is the help button. I just said there's 100 modules in there. Across all the settings, there's about 1,000 settings. We do not expect you to memorize that. I've been using CellProfiler for about 15 years now, and there's still things that I'm like, oh wait, CellProfiler does that? Oh, I had no idea. So we want you to not get lost. We want you to understand exactly what you're doing and why you're doing it. So every module and every single one of those 1,000 settings has help text written by a biologist. So you can always see what's going on and why. CellProfiler has 2 different modes. The one that you'll get the most face time with is test mode, where you go in and then you actually sort of play around and mess with the settings and check that your negative control and your positive control are being treated the same way, because again, you did controls. And then once you've done that, you can go out of test mode, you can set where you want the output to go, and then you can hit this magic analyze button. And CellProfiler will analyze, again, however many images you have, whether that's 3 or 3 million. And then you can leave. You can go to the pub or go to get some free pizza or wherever it is you like to do with your day. And so that's what CellProfiler will allow you to do, and why it might be a good tool for some of you to do the analysis you want to do. This is what the test mode looks like, just to give you a tiny bit more tour of the interface, and then I promise we're going to stop because I know this is not that exciting. You can add, subtract, or reorder modules, and again, there's no limit on how many you can do. I think my personal record is something like 150, but this sort of 10-ish is way more common. You usually don't need 150 unless you're doing something very weird. In this interface, the next module that CellProfiler is going to run, the next thing in the steps, is going to have this green arrow next to it, and it's going to be highlighted and bolded and underlined. We're going to try and make it as obvious as you can what's going on. If you hit step, you're going to run just that one module that's selected and run just that one thing. If the next module, but it will only run the next thing that has a checkbox next to it. And this is actually really nice for if you're testing, if, for example, like if denoising helps your analysis or not, you can put it in and then you can sort of turn it off and see how the results change. Makes it easy to sort of remove something without fully deleting it and making it easy to come back to. once you can look at individual steps, or you can say, I want to see how this whole set of things goes together. Maybe I change my cropping parameters, and then I want to see how that changes my object identification parameters downstream. And if I hit run, it will go all the way until it hits this pause button if it starts from here. Sometimes you want to see what's happening and sometimes not. So there's an eyeball you can open and close, so you can sort of have lots and lots of displays or very few displays. And as much as we can, we try to warn you and tell you, is this set correctly? Is there an error, or is there just a warning? So for example, this is a warning saying, this is a module that spits out spreadsheets of measurements. It's not going to run in test mode because you're in test mode and we assume you're not ready for your spreadsheets of measurements yet. And if you hover over all these things, it will give you that information. There's a workspace viewer, which I won't go into, but it allows you to actually look at lots of images and lots of objects and even their measurements all together in one view. It's a tiny bit crashy, just a little bit, but it's very, very useful. So just save before you open it. And then you start over on the next image set, because again, you're going to remember that you're going to do this on your negative controls and your positive controls. And then once you've done that, you leave test mode, and again, you hit that magic analyze button. There's lots of different kinds of things you can do. There's file processing, there's measurement. I'm not going to read to you. I am going to point out to you that all of this is searchable. So if you search, if you know that what you want to do is to find the size of your cells, you search size here, you're going to find there's a module called measure object size and shape. If you had searched area instead, you're not going to get that module, but you can, if it's not in the module name, you can hit search and it will search all the module help. So that word area is in the help for measure object size and shape, because that's one of the things that it measures. So we're trying to make it as easy as we can to give you lots of options to lots of things, but let you still find the one thing that is the thing that you want. This slide has a ton of text. Don't bother reading it. What I'm going to just tell you is the hardest part about making something that works for time points and multiple channels up to hundreds of channels and lots of Z-planes and things like that is actually you telling CellProfiler what's going on in your experiment, because all of your experiments, I guarantee, look super different. And you have to do some bookkeeping to tell CellProfiler how many Z-planes you have, how many time points you have, how are things arranged in file or in folders. Is like one channel per folder, or do you have one giant CZI file that has all of your data from all of your samples in just one file? You're going to need to do some bookkeeping. This is where people sort of fall off before they even actually get started. It sucks. It's not fun. Nobody likes it. But unfortunately, it needs to be done. We have a tutorial that has a linked video thing of like the top 10 most common kinds of experiments people want to do and how to set these modules up so that you can do them. Again, it's got timestamped video and a written version. And if you have trouble, there's going to be lots of things about how to get help at the end. I know this part's going to suck, and I apologize in advance. It's as least worse as we figured out how to make it. If you have an idea how to make it even less worse, come talk to me. I just want to briefly mention this idea, because this idea is going to be important for whether you're actually going to be using CellProfiler or whatever image analysis tool you're going to do, if you're going to be doing segmentation or finding objects in it. Even if you're doing deep learning pretends you're not doing this, but you're actually doing this. All segmentation works off of the principle, the thing you care about is bright and everything else is dark. Even the things that pretend that they're not doing that are doing that. And so in order to do that, it is fine to do transformations on your images. You can do any transformation you can think of, and that might mean doing something like elastic pixel classifier, or there's lots of other things you might do to transform your image so it looks like that. This does not mean you can then measure that image that you might have done crazy stuff to. Please do not do that. Don't be like, Beth told me I can do whatever I want and I can measure these images that I did crazy things to. But the reason that there are all these image transformation things in CellProfiler and Imagery and all these other places is for segmentation purposes, do whatever you need to do to make the thing I care about is bright and everything else is dark, the truth. So that might mean enhancing dim signal, masking out bright signal that's there because, crud happens. But make sure that whatever tool you're using, whatever you're doing, that to make it so that this is true, that you check your raw data against your segmentations and make sure that you haven't introduced something, whether it's with a neural network or whether it's with sort of a rolling background rolling ball background subtraction, that what it was there at the end really was there at the beginning. Because again, it's not a fun day when you have to go to your boss and be like, yeah, it turns out that wasn't real. All right. So if this were a CellProfiler workshop, now I would start teaching you the principles of classical segmentation. This is just not even quite all of the slides that I would be using to do that. It's a lot of slides. It's like a lot of slides. So one of the things that we do is we try to make CellProfiler better. We try to make doing things the classical way, which is still in a lot of times the best way, better. But we're also trying to find ways where, in the cases where deep learning does add value, which is not every place, but is many places, to make it easier for you to do that instead. One of the good things about ChatGPT is I no longer have to ask you guys if you've heard of deep learning. I'm pretty sure you've all now heard of deep learning. It is not just for summarizing emails from your mom. It is actually also for doing things with images and doing things scientifically. And one of the sort of great things that it's done in bioimage analysis is allowing us to do things like do batch effect correction. Like, let's make today's experiment look like last week's experiment. Do virtual staining. What would my staining have looked like if I did this stain, but I didn't? But where it's gained the most adoption is with cell segmentation, finding things, and not just cells, but really any object segmentation. This is where it's being used the most, and this is where it is the least dangerous to use it. Everything else here is pretty dangerous. This is pretty safe. And there's already, in the last few years, been a tremendous number of really good tools that have come out for doing this. One of them is Stardust, which there's a 2D version and a 3D version. Those of you who work on neurons, I apologize, this isn't good for you because the objects have to be not too weirdly shaped. But the rest of you, Stardust is a great option. CellPose is actually my very favorite software that isn't a software that we make. It's really great. And ever since CellPose 2, it's also retrainable. So you can have it start from a model that they've already made and then give it your data and fit it exactly to your data. We have a CellProfiler plugin if you make a CellPose model and then you want to use CellProfiler for things like measurements. But CellPose is basically black magic. Unfortunately, here's the problem. So we do a survey of the field every couple of years and say, what are you doing? How are you spending your time? Are you doing mostly imaging, mostly image analysis, a mixture? And we also ask people, among a sort of whole other list of questions, how would you rate your comfort in developing new computational skills? So not how much skill do you already have, but how comfortable are you learning something new? It might not surprise you to learn that people who spend most of their time doing image analysis and therefore most of their day on their laptop have a lot more comfort learning new computational things than people who don't. Unfortunately, what that ends up meaning is that the people who really deeply understand the biology, who are more likely to be spending more of their time imaging, are much less likely to be computationally comfortable than somebody who's going to be going in and making a deep learning model. Again, there are absolutely really, really computationally comfortable and qualified people in this bin of doing entirely imaging, but on average, the amount of comfort is going to be less. This is a problem. And this is unfortunately a problem that's only going to get worse. And that's because installing deep learning is, for technical reasons, really freaking hard. This isn't just my team is not good at their jobs. We hire really smart people who are actually really good at their jobs. This is just technologically hard, and it gets harder every time Mac puts out another frickin' new chip. In fact, it is such a problem that there is a central help forum called forum.image.sc, which I'm going to talk about again at the end. If you remember literally nothing else today, remember forum.image.sc. It's a central help forum for lots of open source image analysis tools. And the word install comes up in the first post about 8% of the time. And it comes up in the thread about 1 in 5 threads. So installation is hard already, and it's only going to get worse the more and more the field moves to deep learning. So this is not a small problem. So one of the things that we're excited about potentially doing with a lot of our tools is just making them websites instead. So then we don't actually have to install anything on your computer. You just visit a website and you do all of the computation on the website. And that means that, if there's an update, we can push an update to you right away. You can potentially pull from globally available resources. I understand that a lot of you guys are now doing your image analysis on VMs, which are not actually physically in Cambridge, and that's great. So you're already sort of used to this idea. It means that it's just way easier for you to for us to not have to support you doing stuff on your laptop and you to try to troubleshoot doing stuff on your laptop. And so that was all the inspiration behind this tool we've made called Piximi. You can visit it at piximi.app. If I see you on your phones for the rest of this talk, I'm going to assume it's because you're visiting piximi.app. I assume that's why you're on your phones. What Piximi is designed to do is to allow you to access deep learning in your web browser. Very importantly, except under certain conditions that we warn you about, your data is never leaving your computer. It's not actually going to a central server in somewhere like Holyoke. It is actually staying on your local machine. You can open up the website and put your computer in airplane mode, and except for one or two features that warn you, hey, we're about to send your data somewhere, are you sure? It will work exactly as well with your computer in airplane mode as it works with your computer with its internet connection on. So this is not sending us your data. We're not looking at your data. We're not really interested in looking at your data unless you want to come talk to us about your data. This is all happening locally, but it's happening in your browser without you installing anything. You just visited a website. And so our goal is that with Piximi, that we can support the whole image analysis lifecycle, from you uploading your images to you answering your questions and then being able to just go back to doing whatever science you want to do. So to support that, we have things like where you can draw in your cells, and eventually we will have trainable deep learning segmentation models in there. Right now, we only have pre-trained models, so things like CellProfiler you can do in there. Eventually, you'll be able to train your own. But, if you want to just draw in all your cells by hand, you can draw in all your cells by hand. We have some pretty tools for doing that. Some super pixel tools where you can just click and there's lots of fun hotkeys. Okay, this one does have the color annotation tool. This one's my favorite. This is color annotation. You just click and drag and then you hit enter, and then you can just go through and annotate a whole image really quickly. It's very nice. We can do segmentation models. So here we're going to actually do CellProfiler, which is one of the cases. And so that's actually warning you, hey, this is now going to go to the cloud. So in some cases it will go to the cloud, but it will warn you first. But most of our segmentation models, we have things like Stardust, which are going to happen locally. And so this is all a real-time thing. And this is now showing that it has sent the data to Sweden. It has gone through, it has found cells. The data has now come back from Sweden. And if we look at what these cells look like, they look pretty good. And again, no installation required. And classification is one of actually the earliest things we finish, and it works really well. So here we have cells that have no GFP, nuclear GFP, or cytoplasmic GFP. And a person has gone through and sort of assigned classes to about 60 of those each. And now in real time, this is real time on my MacBook, I'm training a deep learning network that is going to learn to tell the difference between those three things. And it's going to do it with 90% accuracy in the amount of time that I couldn't even finish telling you about it because the slide looped, because that's how fast this happens. So you're going to have the ability to use some of this deep learning stuff without having to learn to code. So for the cases where deep learning adds value, our goal is that you can do it and not have to worry about the computer science underneath. We have computer scientists for that. And you don't need incredibly powerful hardware. This is me doing that same exact classification on my cell phone. It's not as fast on my cell phone as it is when I run it on my MacBook, but it can be done on a cell phone. I have been told, though, it is not cool if you do that at a bar just to show people how cool your software is. I've been told that's not actually very cool, but you can do it. It can be done. And because, again, ultimately, the thing we usually want to do is not just find objects, but know things about them. Sometimes you just want counts, but more of the time you want to actually know things. This feature is right now only available in beta, but I think it's coming over to the main app like today or tomorrow, where you can put in measurements, and then graphing will come after that, where you can then actually make graphs of all of the measurements and sort of split them by things like, what treatment group are they and stuff like that. So again, the whole life cycle all the way through to the answer to your question. Because after all, we don't usually care just about, again, finding an object or doing a pre-processing for the sake of doing it. We want to know something about our data. We want to answer some question. And that is the reason to do software, because we can do lots of measurements. Also because our eyes are really bad at this job, but and because then we can go to the bar. But also because we want to measure stuff. And software, including CellProfiler, but again, lots and lots of tools, are going to help us to build up metrics so we can actually know stuff about our biology. And unfortunately, the vast majority of the time, now this paper is 10 years old, but I'm not sure if we redid the analysis today, it would be that different. Most of the time, I started off by telling you billions and trillions of pixels, and most of the time people report one measurement, two measurements, not very many measurements, which is a shame when you have trillions of pixels to then only look at maybe cell size or cell intensity. And that really inspired Anne and Shantanu, the other PIs in the lab, to really say, how can we use more of those pixels to actually answer other questions? Because there are a lot of different measurements that we can make, even beyond these individual pixels themselves, that, are useful measurements. So probably many of you are used to the idea of, you have some fluorescent label for a thing that you care about. You want to measure if the intensity went up, maybe there's more of the thing, or maybe where the intensity is a little bit different. That's a metric that biologists tend to be used to thinking about. We're also pretty used to thinking about like the size and the shape of the things that we measure. Or colocalization, which, be careful with colocalization. It can mean a lot of things. And it's really a hard measurement to do correctly. So I recommend you talk to the very excellent people who work here, who've organized this talk, and who know a lot about this stuff. But they can actually help you figure out what is actual colocalization and what is interaction. But again, the other thing that I said at the beginning of the talk and then totally stopped talking about is the fact that our brains are real bad at this problem. These are imaging flow cytometer images of immune cells and different cell cycle phases. Who would like to come up and annotate all of these for me? Only once ever in a talk gotten somebody to say yes to that. I was very, very surprised when that person did, because it was on about the 40th time asking. You might have guessed that these are in order by the fact that sort of towards the end they're dividing. And certainly I can tell this one apart from the other ones, but if you scrambled up, especially these first four, I don't think I could reliably unscramble them. And I don't think probably most of you could either, because human brains don't actually know how to do that, because there's texture information that our brains literally do not see. It's not available to us, but it's available to computers. And so here, there was DAPI in these images, what you're not looking at, which was used to determine the actual cell cycle phase. And then the computer was given only the bright field channel and said, predict what cell cycle phase the cell is in. And the computer's doing a pretty much better job than I would do. It's doing a pretty good job. And even the mistakes that it's making, it's not saying that, S is telophase. It's saying, I'm not sure sometimes if an S phase cell is actually G1 or is actually G2. And those of you who've studied DNA replication know these are not perfectly switch-like things. We are forcing biology to be in incredibly tight, neat little boxes, which is not where biology likes to live. So even the mistakes that the computer's making are mistakes that make sense. So there's information here. We can't see it, but it's there. So we can make metrics that help the computer do this. We can make metrics like Zernike polynomials. If I tell you that your treatment caused your cell to get twice as big, you probably have a mental picture in your head. If I tell you that your treatment caused your cell's Zernike 04 polynomial to get twice as big, if you get a picture in your head, congratulations. I do not get one in mine. But which is not to say these things aren't useful. They can tell us about an object's shape, not perfectly, but way more reliably than just if I said, give me the perimeter and the orientation. These are not perfect reconstructions, but they have information in them. And we can measure things like texture. Again, if I tell you that your cell's info measure 45 degrees, pixel value 3 went up, no picture. But we can find out really interesting things like those cell cycle phase measurements based on texture. But again, if we can't use them in the end because they don't make any freaking sense, why am I telling you this? Why am I wasting your time? And that gets us into the last part of the talk, which is about image-based profiling. It's about using all of those pixels. Because now, in the era of single-cell sequencing, probably most of you are comfortable and familiar with the idea of we're going to measure lots of cells and we're going to measure lots of stuff in the cells and then cluster them. You've seen a million UMaps on Twitter or wherever kids are hanging out these days that's not Twitter. But you can do the exact same thing, not just for single-cell gene measurements, but for image measurements, single-cell image measurements. And in fact, you've been able to do that at a single-cell level for about 400 years longer than you could do that on a sequencer. So we can do image-based profiling in the same way we can do single-cell RNA-seq profiling and actually learn things that don't happen at the transcriptomic level, because lots of biology does happen at the transcriptomic level, but a lot of it also doesn't. So this is one of my favorite papers from earlier in the field. They have a bunch of drugs with known mechanisms of action. They have a DNA marker, an actin marker, and a tubulin marker. And they want to know, based on image measurements, can you put the drugs back together into classes? And you're probably not incredibly wowed by 67% accuracy until I tell you this is just with the nucleus, not with the whole cell. So we're seeing perfect classification of protein synthesis inhibitors just in the nucleus. When we go to 3 channels, 94% accurate. And even the mistakes, again, DNA damage and DNA replication are incredibly interlinked biology. So the mistakes are just as much about the tight little boxes that don't actually work in biology as it is about the computer doing something wrong. This is work that then eventually led to a pharmaceutical company that my boss is on the board. I am not on the board. I'm not in any way associated, but just disclaimer, disclaimer, disclaimer. Here's a phenotype that it really looks like humans ought to be able to score. This is the phenotype that you get when you knock down a gene involved in cerebrovascular malformation, which is a disorder that gives you strokes. And so Chris Gibson, who was then at University of Utah, what he wanted to do was screen drugs to say, hey, can we help these people who are getting strokes not have strokes anymore? And this looks like a human scoreable phenotype. If you scramble these cells up, I'm good. I can unscramble those. And so what they did is exactly that. They took knockdown treated cells and they gave them drugs, and then they had a team of human experts pick what are the drugs that make the cells look the most healthy, and they had the computer using CellProfiler measurements do the same thing, and then they made 2 drug lists, human drugs and computer drugs, and they gave the drugs to mice. If you are a mouse, you totally want the computer drugs, because it turns out that even though we can tell the difference, we don't pick up the right kinds of differences. These cells are different in a lot of different ways. You know, things like the shape and things like where this green, I don't even know what's staining green here, is, but we don't pick up on the right one, whatever it is. And so the mice do much better when you give them drugs that were picked out by the computer saying the cell looks healthy. And so about 15 years ago at Broad, they started working on an assay called the cell painting assay, where the idea was essentially if you have a standard 4-channel microscope and as many stains as you can physically shove into the spectrum, and you want to do this as cheap as you can, how many organelles can you see at a time? And it turns out the answer is sort of 8. There is some spectral bleed through. We live with it because we have spatial separation, but probably many of these dyes are familiar to you. You've maybe even used 1 or 2 of them before. And when I first started working in the Broad about 8 years ago, and Ann told me about this thing, I was like, there's no way you learn anything from like organelle dyes. Like, I spent years in grad school making fluorescent protein fusions. Like, no, you need to know where the protein is. You can't tell anything from an organelle dye. Yeah, I was wrong. Not going to belabor this, but it turns out I will assume most of you are not RAF-ROS pathway biologists. I am not either. But what you probably can see, even if not, is that wild types that are identical cluster together when we look at cells in cell painting measurement space, and that the cancer-causing mutations of these things that are all in the RAF-RAS pathway cluster not with these things, but right next to them. So in cell painting feature space, we can already tell cancer-causing mutations from just wild type genes when they're overexpressed. And Mohammed, who now has his own lab, went through and found some pathway interactions that were not predicted by the literature. And we built a reporter gene, and turns out that we discovered novel pathway inhibition from organelle dyes. And I was just like, holy crap, this thing actually works, and this actually lets you learn biology. And so we've done a lot with that in the last few years. I can't unscramble these drug sensitive versus drug resistant cells myself at all, but CellProfiler can based on cell painting features. You can obviously imagine why it might be useful to be able to tell for lots of different drugs at once if cells are resistant to them. You can obviously see the benefit there. We're starting to do things where we can look at cell painting features in iPSC cells from people and say, we're gonna do whole genome sequences, and then we're gonna pull out their SNPs, and then we're gonna pull out their cell painting features and see what's different. And it turns out when you have like thousands of SNPs and thousands of features and 300 people, getting statistical significance is real hard. But we found some things that were statistically significant. And when we CRISPR knock out those mutations that are thought to be inhibiting mutations, we see exactly the same effects that we expected to see in the CRISPR knockouts. In things that are, in this case, possible for humans to see. So 2 of these 3 measurements are human invisible. One of them is human visible, but also pretty hard. If you tell somebody, look at the area closest to the nucleus and see if the mitochondria are symmetric or asymmetrically distributed right around the nucleus, many of you could do that task, and my boss can get these exactly right when we've asked her to unscramble them. But you would have to know to do that, and it wouldn't necessarily be obvious by eye that that was the task that you should do to tell a patient from a non-patient. So if you want to play around with cell painting data yourself, we have about a petabyte of it online in the cell painting gallery. It's all CC0. It's all public domain. You can play around with it. If you want to build computer models for cell painting stuff, we would love to talk to you about that. And we think that this assay is useful for a lot of conditions, and we're also happy to talk to you about doing it yourself. This experiment here was done all by grad students hand pipetting things. It's a lot easier to do this experiment if you have a robot, but you don't need a ton of samples. And again, this was all grad students hand pipetting things. They were fantastic hands, both of them. They were really great. We've put up about 117,000 drugs and 10,000 overexpressed and knocked out genes that you can play around with from big data made from 10 different pharma partners all together, making public data, because Ann Carpenter's a wizard. I don't know how she does it. She's fantastic. We have 3 whole genome knockdown screens for doing this in a pooled context. So say, what if we knock down genes and then we actually figure out what is the gene knocked out in individual cells. We have 3 whole genome screens of that you can play with, and I'm happy to talk to folks about the data. And there's a lot more on there that you can play around with. So hopefully, for those of you wondering, there are 10 minutes left. So if that's what you're wondering, there's 10 minutes left. If what you're wondering is, how can you learn how to do this? Where can you go for help? There's lots of places. Our goal is not to put any of these things behind a paywall. Our goal is to make all of these things available and useful to you now. So for the individual tools, there's in-app documentation. The CellProfiler website has lots of examples and tutorials that will help you get started, like that input module tutorial I mentioned. You can also just look at our examples and see which of these kind of looks like your data and download it and start from there. There's manuals for things. Piximi, we have a Jupyter book documentation, which lets us do fun things like, do drop downs and notes in the margins, so that if you want to learn something, you can, but we're not just going to like throw every piece of information about deep learning at you all at once. If you want to learn about the cell painting stuff, the best place is in the image-based profiling handbook, where we talk through all of the downstream analysis and we link to the open source protocol papers for doing the wet lab side. And we even have a protocol paper for, you've done cell painting and now you have measurements. Now what? Because it turns out a lot of people have that question. So we wrote a protocol for that too. And no matter what kind of image analysis you're doing, no matter what your images look like, we recently put out a open access paper in Journal of Microscopy about common microscopy problems and common solutions and sort of best practices on how to think through just sort of how to set up your image analysis in general, and how it interacts with things like your sample preparation and your imaging. We created this resource called bioimagingguide.org, which is designed to be your introduction to what you don't know about bioimaging and bioimage analysis. So there's information on sample prep, there's information on analysis. Because it's in Jupyter Book, we can, again, do things like we can sort of give you only the information that you want. We can put information for you in the margin that if you want to read, great. And you don't want to read, fine. This is also now translated into Spanish, Portuguese, and Czech, and we're working on 7 more languages. If you have collaborators for whom English is not their most comfortable language, we had actual native Spanish speakers go through and translate, and again, same with the other languages I mentioned. Remember, literally no other thing. Forum.image.sc is where all of these open source image analysis softwares have one central help forum. So you can say, here's my image, here's my question. Somebody help, please. And you get experts from 60 different tools. We've anecdotally heard people are really scared to do this. I swear we're really friendly. We are all huge dorks who want to talk to you about this. Nothing makes us happier than helping you solve your problems and helping actually be useful. So please don't be scared. Please come on and ask us questions. And you can also find conferences and jobs. So if you want to get more into bioimage analysis, this is a good way to do it. And you can come talk to us. We work with groups inside the Broad and outside the Broad. We have tons of collaborators here at Harvard. We're always happy to work with people. We can usually do small projects for free or very cheap, and then we can sort of talk about bigger collaborations as well. And a great place to start that conversation is at our office hours, which we have every 2 to 3 weeks. And so with that, I want to thank all the awesome folks in my group and the Carpenter Singh Lab who did all of this work and who sort of make all of these things actually work for biologists. And thanks, guys, for your attention.